The overall goal of this procedure is to follow the intracellular trafficking of amyloid precursor protein, or APP, in live cells. This is accomplished by first transfecting cells with APP tagged with photoactivatable GFP and compartment markers for lysosomes and the Golgi. Next, the cells are photoactivated in specific regions of interest in the Golgi. Then photoactivation is stopped, and the clearance of APP from downstream compartments is imaged. Finally, trafficking of APP is analyzed using analysis software such as Imaris. Ultimately, photoactivation and confocal imaging are used to show the clearance of APP protein in live cells.
Demonstrating this technique will be Joshua Tam, a PhD student from my laboratory.
After plating SN56 cells and allowing them to proliferate according to the text protocol, in a cell culture hood, use plasmids expressing a fluorescent protein-tagged galactosyltransferase, a fluorescent protein-tagged compartment marker, and a protein of interest tagged with PAGFP to transfect the cells. Incubate the transfected cells at 37 degrees Celsius in a 5% carbon dioxide incubator for 24 hours. To prepare the microscope stage, pre-warm PBS and HBSS to 37 degrees Celsius. Warm up the microscope stage to 37 degrees Celsius. Remove the differentiation medium by using the PBS to wash the cells twice and add 2 milliliters of pre-warmed HBSS. Then place the cells on the warmed microscope stage and allow 5 to 10 minutes for the confocal plate temperature to equilibrate with the microscope stage. With the microscope eyepiece, find cells transfected with Gal-T CFP.